So far we have looked at the role of glucose and glycolytic pathway as a source of energy. However, it's another important function, a source of precursors necessary for other important metabolic pathways. And in this part, we will showcase this role looking at the branch of glycolytic pathway called pentose phosphate pathway. The main branch of pentose phosphate pathway is resulting in the production of pentoses, specifically ribose 5-phosphate, which is used as a precursor for synthesis of nucleotides and various redox cofactors. In this branch of pentose phosphate pathway, starting from glucose 6-phosphate, there are two consequent oxidation steps which at the end result in the release of carbon dioxide from this hexose molecules, thus removing one of the carbon and converting this into pentoses, which at the end becomes ribose 5-phosphate. Because of this oxidation, net oxidation flavor, this branch of pentose phosphate pathway is called oxidative phase. Important byproduct of the oxidative phase is the flavor of reduced flavor of the electron transport carrier called NADP. So it is phosphorylated form of NAD. So in these two consequent oxidation steps, the two reducing equivalents in the form of NADPH produced. And NADPH has very specific and important role, several specific and important roles. First, it is important factor in counteracting oxidative stress. It is used to reduce another redox cofactor called glutathione, and glutathione in turn is important to neutralize active oxygen species. On the other hand, NADPH is also important redox cofactor in fatty acid biosynthesis and in preventing oxidative damage of non-saturated lipids. So in this way, the cells which do not primarily need a lot of pentoses as a biosynthetic precursors circularize this pathway, extending it with non-oxidative phase. And in this non-oxidative phase, the pentoses are produced in the middle of oxidative phase are undergoing a series of backbone rearrangements such that several of these pentose molecules, 5 actually in, sorry, 6 in total, give rise, get recycled back to 5 glucose 6-phosphate molecules. And again, this pathway as such would be would produce would not produce any any downstream metabolites, but yield only the reducing equivalents in the form of NADPH and carbon dioxide. Imaginably, these pathways, this flavor of pentose phosphate pathway, are predominant in different kind of tissues depending on where which flavor is needed most. In highly proliferating tissues, like in epithelial cells or in immune system, the pentose phosphate pathway is mainly an oxidative phase, which is important for production and a lot of precursor for nucleotide biosynthesis necessary for cell proliferation. But in non-dividing tissues which are susceptible to oxidative stress, like in um, erythrocytes or in liver tissue or in cornea, which is directly contacting oxygen-rich environment, the non-oxidative phase is flavor is predominant, and yeah, similar to the adipose tissue, for example, where it is practical for lipid biosynthesis. But let's start with oxidative phase. Oxidative phase consists of several reactions again, and two of them are oxidations. In the first oxidation step, the enzyme called glucose 6-phosphate dehydrogenase uses NADP as a cofactor to remove the electrons and convert the circular form of glucose 6-phosphate into intermolecular ester, essentially in the anomeric carbon of glucose 6-phosphate is oxidized given this lactone molecule, 6-phosphoglucono-gamma-lactone. And this is followed by linearizing this circular ester by lactonase, given this the carbonic acid 6-phosphogluconate, which is in turn oxidized once again by phosphogluconate dehydrogenase, again using ADP plus as a cofactor, and this results releasing two more electrons in the form of an ADPH and breaking this backbone, releasing the terminal carbon in the form of carbon dioxide, which results in this ketose diribose, diribulose 5-phosphate, which in turn is isomerized by phosphopentose isomerase into diribose 5-phosphate, which is straight precursor for nucleotide and nucleotide-based cofactor biosynthesis, redox cofactor biosynthesis. But when the NADPH is a product which is primarily necessary, then this again pathway is branches off at the level of ribulose 5-phosphate, which is epimerized, forming deoxylulose 5-phosphate. So it is epimer of diribulose 5-phosphate, where one of the chiral centers is flipped. And this originates a series of interconversions where the enzymes transaldolase and transketalase are used to disproportionate or transfer the carbon pieces of various sizes, carbon skeleton pieces of various sizes between the two flavors of pentose. And this is quite a complex chain of reactions where first two carbon pieces are brought from one pentose to another, this way given one hexose and one triose, and then heptose and triose again undergo an exchange in the carbon backbone given hexose and tetrose. Tetrose exchanges with another pentose given again hexose and triose is at some stage get joined back into hexose by aldolase, which is shared enzyme between non-oxidative phase and the glycolytic gluconeogenic pathway. So this complex chain of backbone exchanges results in total in formation of five glucose 6-phosphate molecules out of starting six the pentose molecules. While these backbone rearrangements are highly reversible reactions, the main oxidative phase of pentose phosphate pathway is essentially irreversible. Both oxidation steps are irreversible reactions. This raises the questions how cell regulates partitioning of the metabolite flux between glycolysis and the pentose phosphate pathway. And the important role here is played by the enzymes themselves, which catalyze respective steps in glycolysis and pentose phosphate pathway, which turn out to be regulatory enzymes. So one important regulator of this partitioning is the first enzyme in pentose phosphate pathway, glucose phosphate dehydrogenase. This enzyme is highly allosterically regulated by NADP+. NADP+ signals the shortage in NADPH, and it activates it works as a potent activator of glucose 6-phosphate dehydrogenase. This way, if cell is in the need of pentose phosphate pathway, this enzyme is activated, and then the flux of glucose 6-phosphate is diverted to the glucose 6-phosphate pathway. However, if the NADPH is in excess, it would signal that this pathway is saturated, this enzyme get inactive, and then main flux is diverted to glycolysis and ATP production. So this is only part of the complex regulation network of these pathways, but it shows the point. So this part concludes our excursion into glycolysis pathways and glycolysis pathway and its flavors.